Hello everyone, this is my presentation on the human immunodeficiency virus, or HIV, and its testing methods. So the structure of HIV is an enveloped capsid, and within that capsid is RNA with polymerase, gene proteins, reverse transcriptase, protease, and integrase, which are important for its proliferation within the cell. And the envelope glycoproteins 120 and 41, and the capsid protein 24 are very important. So the main target of HIV is T helper lymphocytes, or CD4 T cells. Dendritic cells are the first to be infected, but because they are antigen-presenting cells, they will present HIV to the CD4 T helper cell. And the glycoprotein 120, which 120 is just the molecular weight in kilodaltons, will attach to the CD4 and then will change shape and via the V3 loop attached to a co-receptor CXCR4, which is a protein found in a plethora of cells. And this will expose GP41, or glycoprotein 41, to release the capsid into the cell. Once the capsid is in the cytoplasm, the cellular enzymes will strip away the capsomeres, releasing the HIV genome. P24 coats that genome. And once it is released, reverse transcriptase will transcribe RNA into DNA and use the newly formed singular DNA strand to create its complementary strand, which then migrates into the nucleus. The use of reverse transcriptase in cellular biochemistry is very new, considering most cellular processes use DNA to create RNA. This reverse process is why HIV is considered a retrovirus. Via integrase, the viral DNA will become part of the host DNA, turning it into a provirus, and this can either be latent, which it means it does not express, or it can express, causing the cells to become a viral component factory. As the viral components assemble just inside the cell membrane, they will take some of that membrane with it as they lyse the cell, causing the CD4 T helper cells to dwindle to a point below 200 cells per microliter, causing the disease to transition into autoimmune deficiency syndrome, or AIDS. Testing methods have vastly improved since the 1980s. Nucleic acid testing was the first line test, but it can show false negatives due to the eclipse period, which is the time between an infection and the production of antibodies. So it's usually done after diagnosis for better prognosis of the patient by detecting viral RNA. Current immunoassays test for both HIV-1 and 2, but may lack specificity, so they incorporate anti-P24 antibodies to detect P24 antigen, which is a protein that coats the RNA. Other testing methods include rapid testing, which is great for places that don't have advanced technology for testing but aren't very reliable. Western blot used to be the golden test, but is not very time sensitive, so it's not really used anymore. Reverse transcriptase PCR is usually always very reliable, but it's not very time sensitive either. Here are my references, and I hope you enjoyed the presentation.